So in the previous video, we have seen in general what are reactive oxygen species. So we'll see in detail each. So first one is hydroxyl radical. So as we know, it is extremely reactive, and immediately removes electron from molecule and turning that molecule into a free radical. So thus propagating a chain reaction. So this is a very reactive since it undergo a chain reactions. It is very reactive, and we can prepare this hydroxyl radical to two methods. First one is Haber-Weiss reaction. There oxygen like superoxide on reacting with the peroxide in presence of iron or copper ions, we will get one OH and OH hydroxyl radical, hydroxyl ion, and oxygen. So that is the first one preparation method. Then secondly, it comes Fenton reaction. Fenton reaction, as we have seen in previous videos, I think this was once we discussed this during the carbohydrates and all. So Fenton's reaction is hydrogen peroxide with iron or copper. Like this is the Fenton's reagent. We'll get a free radical, hydroxyl radical, and a hydroxyl anion, and this will be oxidized. Fe2 plus will be get oxidized to Fe3 plus. So hydrogen peroxide. So this is about hydroxyl radical. This hydrogen peroxide in both the synthesis method, either in Haber-Weiss and Fenton reaction, we used hydrogen peroxide. This hydrogen peroxide is actually more hazardous or more than it. This hydrogen peroxide damages more the DNA than this hydroxyl ion, since the low reactivity of hydrogen peroxide provides enough time for molecules to travel into the nucleus of cells, subsequently reacting with the macromolecules. Like, okay, since as hydrogen peroxide is actually more reactive or more dangerous to DNA. Why to DNA? Because the reactivity is actually lower. It won't react quickly. The reactivity of hydrogen peroxide is actually low, so it can travel. It will get enough time to travel into the nucleus of the cell and therefore damaging macroscopic molecules. Then some comes the singlet oxygen. It is also an reactive oxygen species. So singlet oxygen, it is produced as a byproduct of photosynthesis in plants. So in plants, due to the excess light, increased production of CO2, it results in cell death. Okay, in presence of light and oxygen, photosensitors convert this oxygen to singlet oxygen. Okay, so this generally this triplet oxygen is getting converted to singlet oxygen in presence of light and a photosensitizer. In, of course, in plant, the photosensitizer will be the chlorophyll. So this is highly reactive. Singlet oxygen is highly reactive, especially with organic compounds that contains double bonds, and this causes damage. Because of the production of singlet oxygen, this reduces the photosynthetic efficiency of chloroplast. Okay, so these are the two main reactive oxygen species. So other than this, we have many other reactive oxygen species. So the one is the carotenoid, plastinoids, and tocophenols. These also contained in chloroplast. Okay, so additionally, we said, like, okay, in plants, this oxygen in presence of light and photosensitizer, that is the chlorophyll, this is getting the or singlet oxygen is produced. Okay, so this singlet oxygen, it is actually hazardous, or this reduces the efficiency of photosynthetic in photosynthesis of chloroplast. This efficiency is reduced actually. So other than the toxicity, this acts as a signaling molecule. Okay, also it acts as a signaling molecule. Also, the point is, this carotenoids, the toxicity level of singlet oxygen can be actually controlled. So carotenoids, tocophenols, plastoquinonin, actually the bond structure is plastoquinonin. So These contained in chloroplast. These quench this singlet oxygen also and protects. The toxic effects of singlet oxygen can be reduced or minimalized. It protects these carotenoids, tocophenol, and plastoquinol. These will protect the or it is not actually protect. This will quench the singlet oxygen and thereby reducing the toxic effect. Okay. Also, oxidized products of beta carotene rising from the presence of the singlet oxygen act as a secondary messenger. So it can either protect against singlet induced toxicity or initiate a cell death, programmed cell death. So actually the singlet oxygen has negative and positive. This production of singlet oxygen also can be controlled by the presence of carotenoids, tocophenols, and plastoquinol. And the oxidized product of beta carotene arising due to the presence of the singlet oxygen act as a secondary messenger. How it act as a secondary messenger means it can either protect against this singlet oxygen induced toxicity, or it can initiate cell death. So we'll see nitrous oxide in the next video.